Good morning. My name is Adele Myers. I'm a physics PhD candidate at UC Santa Barbara in the Geometric Intelligence Lab. I'm also a trainee of the Ann S. Bowers Women's Brain Health Initiative, and today we're going to do a tutorial on geometric approaches for processing brain connectomes. So, what is a brain connectome? Before we answer that question, we should talk about what white matter is. So, what is white matter? White matter is referred to as the highway of the brain because it transports the information. It consists of bundles of axons or tracts, which are the long, slender projections of nerve cells. And, fun fact, unlike gray matter, which peaks in development when we're 11 or 12 years old, white matter keeps developing well into our 20s and perhaps in more subtle ways into our 50s. So, a connectome is a measure of which brain regions are connected. There are two types of connectomes, structural connectomes and functional connectomes. And to think about the difference, we can first assume that the brain is divided into n areas. And then the structural connectome is how the areas are structurally connected. So for example, how many fiber tracts link two areas. The functional connectome, on the other hand, is how the areas are functionally connected. So whether one area co-activates with another area when the brain is shown an image of a cat or something. Do two areas of the brain light up at the same time? And we're going to be focusing on functional connectomes in the tutorial today. So why are we interested in functional connectomes? The running example that we're going to use is diagnosing schizophrenia. Here we see our first image of what a functional connectome could look like, and we see that it can be seen kind of like a matrix. On what could be called the x-axis, we see the 0 through 25 markings. These are the 0 through 25th brain regions that we're interested in measuring. And then on the y-axis, we see another 0 through 25 markings, which are also the exact same 25 brain areas. And when we look at what's in the middle, it shows which brain areas are connected to which other brain areas. So if we look at, for example, the 15th brain area's connection to the 0th brain area, we see kind of a dark square, which means that these two areas are not very connected. But if we look at the 15th brain area's connection to the 17th brain area, we see a lighter square, which means that those two brain areas are more functionally connected. And already just looking visually at the schizophrenic connectome versus the healthy connectome, we see that the healthy brain has a lot more connections than the schizophrenic brain does. So we see that the functional connectome, can be used to distinguish between a healthy brain or a schizophrenic brain. So, to quantify differences between a healthy brain and a schizophrenic brain, we must be able to quantify differences between the healthy brain's connectome and the schizophrenic brain's connectome. So before we were able to visually kind of see what the difference would look like, but how would we numerically quantify the difference between two connectomes? Our answer is that each connectome can be represented as a correlation matrix. Earlier, I said that you can kind of see that the connectome could look like a matrix of some sort. We're actually going to represent it as a correlation matrix. Correlation matrices happen to be symmetric, positive, definite matrices. Luckily, the field of mathematics has developed lots of ways to compare SPD matrices. One common method utilizes the manifold of SPD matrices. When I say a manifold, you can just think of an abstract mathematical space. And in the case of the manifold of SPD matrices, every single point in this abstract mathematical space is a different SPD matrix. So to compare two SPD matrices, we just have to identify which points they correspond to on the manifold and then measure their distance from each other on the manifold. So how do we measure distances on manifolds? This is an entire area of study. We're going to go over a very brief summary, but if you're interested in this topic, I welcome you to check out the tutorial on geomstats, which is linked here. Geomstats is a software package used to compute statistics on manifolds, and this tutorial is meant for complete beginners. So in normal Euclidean space, we can compute the difference between two matrices by subtracting one matrix from the other. However, we cannot use normal Euclidean mathematical operations, such as addition, subtraction, multiplication, division, to compute distances on manifolds. Instead, we must use manifold operations. Now we're going to go over what are the elementary manifold operations. So when you think about operations in Euclidean space, the elements that you're working with are points and vectors. The elementary operations are addition and subtraction. In addition, you take a point, add a vector, and you get another point in the Euclidean space. In subtraction, you take 2 points in the Euclidean space, and the output is a vector. Linear distance quantifies the length of the line between 2 points. The line is the shortest. The line realizes the shortest distance between 2 points in Euclidean space. Manifold operations are similar, but as you'll see in a second, they're a little bit different. For this case, we're going to use the example of a circle, or I guess we've visualized a sphere over here, but specifically in these sub-diagrams, we're using the manifold of a circle. The surface of a sphere is a manifold, and the circle is also a manifold, because like I said, a manifold is just a set of points. So the manifold of the circle is just the set of points that lie along a circle. So on the manifold case, the elements are points that lie on the manifold. So instead of the Euclidean case where we can consider points anywhere in 3D space, we have to consider points on the manifold. And then instead of vectors, we have tangent vectors on the manifold. A tangent vector is defined at a specific point on the manifold and is tangent to the manifold at that point. Then for operations, instead of addition and subtraction, we have the exponential map and the logarithm map. The exponential, similarly to addition, takes a point and a tangent vector at that point and gives you another point on the manifold. The logarithm takes 2 points on the manifold and gives you a tangent vector at one of those points. And the geodesic distance gives you the length of a geodesic between 2 points. Similarly to how a line realizes the shortest distance between 2 points in Euclidean space, a geodesic realizes the shortest distance between 2 points on a manifold. So how are we going to use this? We're going to eventually be using this for machine learning on manifolds. So before we talk about machine learning on manifolds, we should talk about machine learning in general. So diagnosing schizophrenia in machine learning terms is essentially a matrix classification problem. Classification in general terms learns a function that maps an input X to an output Y based on example input-output pairs. We say the model is a classification if the outputs are categorical, and a regression if the outputs are quantitative. In our case, we're training an AI model to know the difference between a healthy brain and a schizophrenic brain. So when we train the model, we'll give the model a bunch of connectomes, and we'll tell the model whether all of those connectomes are schizophrenic or healthy. And then in the end, we'll be able to show the model an entirely new connectome that it has never seen before, and it will be able to predict whether that connectome is schizophrenic or not. The model we're going to use, the model, is a classification method called minimum distance to mean. We're going to first describe what that is in the normal Euclidean case. So to train the model, the algorithm computes the mean of all the points with the same label for each class. So in our case, the algorithm would compute the mean of all the schizophrenic connectomes and the mean of all the healthy connectomes. And then to test the model, so when the model is shown an entirely new connectome, we would feed the model a connectome with an unknown label, and the model predicts which class it is, whether it's schizophrenic or healthy, by calculating which class mean is closer. And it uses the Euclidean distance to determine which class mean is closer. And the closer class mean is what this new point will be classified as. Minimum distance to mean on manifolds is a similar idea, but in training, instead of learning the mean of each class, we learn the Frechet mean of each class. The Frechet mean is essentially the mean, except it uses manifold operations to compute the mean instead of Euclidean operations. So instead of using addition, we use the exponential map. Instead of using subtraction, we use the logarithm map. In testing, instead of comparing the linear distance between the test point and the class means, we compare the geodesic distance between the test point and the class means. So because connectomes, recall that connectomes we're going to represent them as points on the SPD manifold. So we're going to use minimum distance to mean on manifolds to classify connectomes. I mentioned geomstats earlier. Geomstats is a software package for computing statistics on manifolds developed by the Geometric Intelligence Lab and our collaborators around the world. And we're going to use geomstats in our tutorial because it gives us all of the tools that we need to do what we want to do. So, like I said, this is a tutorial for classifying brain connectomes with minimum distance to mean in geomstats. The data we're going to use in this tutorial, we have 86 connectomes from 86 subjects. Among them, 46 are from healthy controls and 40 are from schizophrenic subjects. So first, we'll import all of the packages we need, including the geomstats packages. We load the connectomes, which in this case, the data has already been uploaded to geomstats, so we load it from geomstats. And before we do any sort of machine learning, it's always good practice just to inspect the data. So the brains have been divided into 28 areas. The connectomes are 28 by 28 matrices that are symmetric. We have, and we're just going to create a figure that shows the first two data points in our connectome data set. This is actually what I showed you earlier when I was introducing functional connectomes. Next, we're going to verify that the connectomes in data actually belong to the manifold of SPD matrices. This is another good practice when you're working with data on manifolds, is just make sure that your data actually lies on the manifold that you think it does. And when we feed the SPD, the connectome data through manifold.belongs, which is a geomstats function that just checks whether the data falls on the manifold that you're considering. It returns true. So here we have created our manifold object. We imported the SPD matrix manifold from geomstats, which we then used to create the manifold object. And then we used this manifold object to determine whether all of the data belongs to the manifold. Now we can start our machine learning. So recall that the general idea of the Riemannian minimum distance to mean classifier is to first train. So we compute the mean of each class in the train set using fresh A mean, and then test, which means give the label. We want the classifier to give the label of the class whose mean is closest to the new data point using metric.dist. And this is just a function that helps us compute distances on manifolds. So this should be, first we create our classifier object because we already created our manifold object a few slides earlier. There are two classes, so that's why we set the classifier.classes function to be 2. And now first we split the data set into a train data set and a test data set. So the train data set is what we're going to use to train the model, and the test data set is what we're going to use to test the model, to see how well it can classify connectomes that it's never seen before. And to train the data set, or to train the model, all we have to do is MDM, which is our classifier object, dot fit, and give it the X and Y. And now we're all ready to test. So to test, what we can do is MDM.score, and it tells you how well this model was able to classify, predict schizophrenia on data it has never seen before. So what if we had disregarded the geometry of the space? What if we had just acted like matrices don't fall on the SPD manifold and just computed everything using Euclidean distance? Well, we can test that using sklearn's nearest centroid algorithm. So first we have to reshape all the data to be compatible with the nearest centroid algorithm, because geomstats can take higher dimensional data than nearest centroid algorithm can. That's actually one of the advantages of geomstats, is it's well designed for higher dimensional data. And then when we train and test, we see a different score. So we see that geometry provides a prediction advantage. With geometry, the score was 0.727, and without geometry, it was a little bit lower. So that concludes our tutorial. In summary, we discussed what is a connectome, why are geometric algorithms useful for analyzing brain connectomes, and how to use geomstats to build a schizophrenia diagnosis tool in Python. Thank you. Thank you for joining, and good luck with whatever project you're working on.